Welcome back to the first Corrosion and Materials Degradation web conference. It's a pleasure to introduce now the next session and the session chairs, Professor Dr. Judit Telegdi and Professor Dr. Daniel John Blackwood. Judit Telegdi, who is Professor Emeritus of the Hungarian Academy of Science, as well as of the Óbuda University, has spent more than 30 years in studying of corrosion, inhibition of corrosion, especially of microbiologically influenced corrosion by inhibitors and by nanolayers, as well as by smart coatings. Her results were published in more than 188 papers and book chapters. Dan Blackwood is an associate professor at the National University of Singapore with 30 years of experience in the field of corrosion. He first became interested in microbiologically influenced corrosion during the early 1990s, while working for the United Kingdom Atomic Energy Authority on corrosion and gas generation in relation to nuclear waste repositories. He has published more than 140 journal papers on corrosion and electrochemistry. Thank you very much to both of you for chairing this session. And now the floor is yours, please. Thank you.
Okay, thank you, Cedric. And hopefully we can maintain the high standard that we've had so far in the conference. I've been impressed by the number of people attending each session. We've got 6 speakers, 2 invited talks and 4 submitted papers. And it's my pleasure to introduce our first invited speaker. That's Professor Dake Tkachuk. He got his PhD from Ohio University, and he's published more than 100 peer-reviewed journals, papers, several in corrosion science. And he's one of the, what they call 10,000 talents, I think it is. Or was it 1,000 talents, which China's trying to pull back to their own country. They had a number of top people all around the world, and they're trying to pull them back to their own country. His research interests are in microbial corrosion, antibacterial material, and biomaterials, biofilm and biocide. So he's basically a cross-disciplinary person from materials science, corrosion, and microbiology. He's the editor of Journal of Material Science and Technology, and he's on the editor board member of several other journals. He's given keynote talks at Eurocore, and he's presently at the Shenyang National Laboratory for Material Science, Northeastern University. Okay, so I will hand it over to you, Dr.
First, appreciate for the introduction by Dan. And it's my great pleasure to take this opportunity to present my current work in MIC. And thank you for the invitation from the MDPI and from Judit. So I just finalized my presentation a couple minutes ago because I plan to introduce some mature work done a year ago, but I changed my mind today because I want to introduce some ongoing work of microbial corrosion caused by geobacterial species. Partially, the work is published, but most of the data are unpublished. I will have 3 parts for my presentation today. First of all, I want to introduce my group of members and myself. I think most of the content I have already introduced by Dan, so I will save some minutes. My background is for my bachelor and master degree, I'm in microbiology. So I learned like molecular biology and microbiology. And I obtained my PhD degree in Corrosion Center in Ohio University. And as you know, it's a famous corrosion institute in oil and gas industry, and where I learned corrosion and bioelectrochemistry. And I started my own work since 2013 and in Institute of Metal Research, Chinese Academy of Science, where I learned the knowledge for materials. And now I'm an independent investigator in Northeastern University. And my major research interests focus in MIC mechanism, MIC detection, and its prevention. And it's my great honor to take a visiting scholarship at Professor Derek Lovelace's group during 2019. As we know, Professor Derek Lovelace is the most famous scientist in extracellular electron transfer and in microbiology. And I have published more than 100 peer-reviewed paper and 20 in corrosion science and 8 in bioelectrochemistry and 8 in IBB. And we just got one paper published in ISME journal, which is the top level journal in microbiology, and it's a Nature series journal. And now we have a quite young group, and it's quite interdisciplinary across the member background. We have Professor Yongqiang Fan as a molecular biology, who focus on synthetic biology to make genetic engineered mutant for MIC mechanism research. We have Messi, who is from Iran, and her background is electrochemistry and corrosion. And we have a new member, Mingxing Zhang, just graduated from University of Virginia, who can set up the newest model LLSM that can be used for single cell level research of microbes or cells. And we have Assistant Professor Zhong Li, who is a microbiologist. And now we have more than 20 PhD students and around 15 master students. I think most of the audience, we are in this area, so I will take a shot for the background. I think, first of all, we do MIC research because of MIC leads to a severe economic loss. 20% of direct economic loss are caused by MIC, and in 2016, we have an investigation in China, and our number of the economic loss in China is 420 billion RMB, and 20% of them are caused by MIC. And we got a news last year that Titanic will be disappeared in recent years. And one of the major reasons is caused by some severe corrosive organisms. And as far as we know, MIC can corrode almost all metal materials, even for copper. As we know, copper is antimicrobial, but like SRB can still corrode copper. And even for titanium, the corrosion problem of titanium is neglected because of its super corrosion resistance, but unfortunately, it is not immune to MIC. And we even do some research in oral microbial corrosion, because as we know, oral is a place rich of biofilms, and there are even electroactive biofilms in oral environment. So I say MIC is ubiquitous. And this is my personal understanding, the existing problem for MIC research. Most of corrosion engineers and researchers, we focus on corrosion behavior evaluation for new materials or some new environments. This is good for industrial application, but it slowly got deeper into the mechanism research. And there's a gap between material people, corrosion people, microbiology people, and electrochemists. So a common language is needed in this area. And the most important thing is we are still lack of MIC mechanism. The whole mechanism is pretty understood. Why we need a mechanism, for my personal understanding, if we have the mechanism, we will do monitoring and detection of MIC, because now we don't have a biomarker, or even we don't have an exact marker for detection and monitoring. And only if we have a mechanism, we will have a guaranteed precision prevention methodology, like targeted prevention methodology for MIC. As we know, we have like LPR-based technique for MIC detection, but unfortunately, it always misleading for the MIC mitigation, because they cannot figure out the difference between organic short film, inorganic short film, and biofilm. So the data obtained by like BioGeorge or other LPR-based technique is not reliable. So that's why we need MIC mechanism. And as we know, since 100 years ago, people start to learn MIC, and many mechanisms are proposed during this 100 of years. But unfortunately, no mechanism can reasonable to explain most of the scenarios in field or in laboratory test. And in recent 10 years, people realize that maybe microorganisms can uptake electrons from metal directly and based on different electron uptake methods. I think my own work is focused on the what we call the extracellular electron transfer between biofilm and the metal. I think this is the key to dismiss MIC mechanism. And based on the last 10 years investigation, we realized that bioenergetics can explain why MIC occurs, and extracellular transfer can actually explain how MIC occur. First of all, our target, we are talking about corrosive microbes, but some of the microbes are electroactive, but maybe some, they just produce like corrosive metabolites like H2S or acidic metabolites. So what we are talking about electron transfer is we focus on EAB. And EAB can serve as a microbial electrocatalyst, but not all microorganisms are electroactive. And extracellular electron transfer is the key link between the solid state electron donor and acceptor to microorganisms. So for us, the electrode, if it is a carbon steel, is a stainless steel, they are the electron donors for microorganisms. So here we have a common language to make a definition that direct electron transfer or mediated electron transfer. I think Dan will raise some concerns for his presentation, but here, what we define the direct electron transfer is if there's no mediator like a riboflavin or phenazine or hydrogen, we call it direct electron transfer by a protein or enzyme. If it rely on riboflavin or phenazine or hydrogen, it's mediated electron transfer. So based on thermodynamics, we can explain why microbes are corrosive, because they want to obtain maintenance energy. So this will be short. And we verify that starved biofilm will cause accelerated MIC. And we confirmed with SRB and NRB for this concept. And also for the research group from University of Akron and the paper published by Dan, they all confirm that when deprived of energy source, such as organic carbon, the microbes will become aggressive because they want to obtain energy, so they want to corrode to make a living. So I define ET can be the method how microbes corrode. So we use ET to explain how MIC occur. ET is has been deeply investigated in microbial fuel cell. And we defined MIC is a biocathode type of microbial fuel cell. That's how microorganisms uptake electrons directly or indirectly from the electrode. And we make a proposal mechanism for SRB. We think the electrons will uptake by SRB via the electron transport chain and result in MIC. And we also verify that the corrosion caused by sulfate-reducing bacteria is mainly due to EET rather than the biogenic H2S at neutral pH. This is published in Corrosion Science. And we also tested that electron mediators will accelerate corrosion of SRB, even promote MIC called by NRB. And then a couple years ago, we moved to MIC research into genetic level because we can make some genetic engineered mutant for deficient or overexpression of actual cellular electron transfer regulated genes. So we first demonstrate that endogenous phenazine encoding genes regulate the ET-MIC caused by Pseudomonas aeruginosa. And now we move to the topic of today. I want to introduce DET caused by Geobacter sulfur reducens. Because before we make many assumptions that electron microbes can corrode via direct electron transfer, but we have no solid evidence or rigorous experiment to demonstrate this. In 2019, Professor Derek Lovely published one paper in MBio, which is a top-level journal in microbiology. They think ion corrosion can be occurred via direct electron transfer caused by Geobacter sulfur reducens. But in this work, I have two concerns. First of all, zero-valent ion they use can be coupled with cathodic hydrogen evolution. And the second concern is based on their transcriptome data, both of hydrogenase and the cytochrome C, the outer membrane cytochrome C, are upregulated. So we cannot confirm the role of this outer membrane cytochromes. They are really responsible for MIC. So when I do my visiting professor there in Derek's group, we make some new mutants and we want to avoid the influence by hydrogen. So we change carbon steel or zero-valent ion into stainless steel, and we make a fresh water medium. So in this medium, fumarate will be the sole electron acceptor, and stainless steel is the sole electron donor. So if MIC occurred, the only electron transfer pathway is Geobacter obtain electrons from stainless steel and then pass it to the fumarate. So we do a hydrogenase deletion mutant, and then we delete the cytochrome S of this string, and then we complement back into the mutant. So this is the biofilm morphology, and we can see based on the hydrogen measurement, when we use stainless steel, there's almost no hydrogen produced. First of all, the OCP cannot satisfy the thermodynamics for cathodic hydrogen evolution. And if we use zero-valent ion, they will produce many hydrogen. So based on this, we eliminate the influence by hydrogenase by use stainless steel and delete the hydrogenase genes. And then we do the MIC test. We can see when we delete the hydrogenase, the Geobacter are still corrosive. But when we delete hydrogenase and then delete the OMCS, they are become very friendly and benign. They will lose their corrosivity. And if we complement back the OMCS genes back into the mutant, they recover their corrosivity again. And then we do the electrochemical measurements. All these data are consistent that OMCS will be play a key role in the electron transfer process, which results in the MIC of Geobacter. And then we do the EIS testing, we got the same data. And as I mentioned, the fumarate will be the only electron acceptor. So we will check the succinate concentration. When fumarate obtain electrons, they will change into succinate. We can see when we delete hydrogenase, there's no significant difference for fumarate reduction. But when we delete OMCS, the fumarate reduction significantly decreased. And if we complement the OMCS gene back, they will recover their corrosivity, so they will recover the reduction of fumarate to succinate. So we first submit this paper to PNAS, and the editor raised a concern. Hey, you just test only one species, so you cannot claim it's a common theory. And then we find another strain. This is called Geobacter motility reducens. And we do the same experiment. We make stainless steel as sole electron donor, and because Geobacter motility reducens cannot reduce fumarate, so we use nitrate as electron acceptor. We repeat the same, we do the same mutant, we delete the OMCS genes. We found the same thing, that when OMCS is deleted, the corrosivity is disappeared. And when we complement it back, the corrosivity recovered. So here we can claim a new theory that, like Geobacter sulfur reducens, they can use outer membrane cytochrome S as a electroactive protein, which can do electron transfer to uptake electrons directly from stainless steel. So if we delete these genes, the microbes will become very friendly and become benign and lose their corrosivity. And this is just published in ISME journal. This is a Nature Theory journal and top level in microbiology. And followed by this, we want to fully verify that OMCS play a key role. So we use nanoparticles to compensate for the lack of OMCS in stainless corrosion. If nanoparticles can compensate this, we can double confirm the direct electron transfer in MIC caused by Geobacter sulfur reducens. The idea we have is we follow some research in ET, and the paper are recently published in Czech. So we buy the iron oxide in size like 50 to 100 nanometer. And this is the initial work, and we have a series work in nanoparticles. So this is just a preliminary data without publication. We first make the nanoparticles attach on the electrode, and then we test the corrosivity. We found that with addition of nanoparticles, it doesn't influence the environment, doesn't influence the corrosion of the sample. Then we do add the Geobacter sulfur reducens. We can see the nanoparticles are well associated with the bacteria. This is the ICM image. And this is the TEM image. We can see without addition of nanoparticles, we can see some pili. And when we add nanoparticles, the nanoparticles are well associated with the cell membranes. And the nanoparticles may even facilitate the production of pili. Then we observe the morphology of the biofilm. We see no significant difference with and without nanoparticles, and even for the pH. Then we do the corrosion evaluation. We found that when the ACL strain, we add nanoparticles, the RP decreased, it means corrosion increased. And when we add nanoparticles with hydrogenase deletion and OMCS deletion mutant, before they lose their corrosivity, but when we add nanoparticles for this yellow color, the corrosivity increased. So this can be verified by the polarization curve. We can see when we add nanoparticles, the corrosivity increased. When we add the nanoparticles, the corrosivity increased. And the same data obtained by the pitting morphology examination, it is highly consistent with the electrochemical data. Oh, I think we also have the weight loss data. So because I just make the presentation, so please forgive me. There's also the weight loss data as fully consistent with our concern. So just now I mentioned pili. So based on the outer membrane morphology research, the most important protein are OMC family. But for Geobacter, they still have another important protein responsible for extracellular electron transfer. This is pili. So we adopt the same idea. We make the mutant of pili deficient mutant to see what's going on for their MSC behavior. So
can you
here we
finish in
can
about
do
2 minutes?
Okay, I will done in 1 minute. we do a TM image. We found that for the wild type, we can clearly see the pili. But when we knock out the pili genes, the Geobacter cells lose their pili production ability. And we do the similar stuff for electrochemical measurements and for weight loss and for piece morphology examination. And we got one conclusion is that PLA getting involved in MSC of Geobacter electron transfer. And then we compare the role, which is more important of OMCS or PLA. So based on the current data we have, there are similar contributor to MSC caused by Geobacter sulfate reducens. So this is our preliminary mechanism. When we have pili, they will have a better direct electron transfer efficiency. When we knock out the PLA, they just rely on OMCS. So their efficiency is decreased. So that's why our mutant show the less corrosivity than the wild type biofilm. And I highly appreciate for the help for my former PhD advisor, for my boss, and for Professor Derek Lovelace, and also all my cooperators for their contribution, and for all my graduate students and my family members. And two short advertisement. We have a journal called Journal of Material Science and Technology. The emerging impact factor this year will reach 7.5 to 8. And I'm the editor responsible for corrosion and antimicrobial material. So it's highly welcome your high quality paper to this journal. And this is free, it's not open access, and we guarantee a good editor service and a good author experience. And we are quite international group, and we are highly welcome for international students and postdocs and even for staff. So thank you for your attention. Thank you for the invitation by Dan and Judit. Thank you.
Thank you. Thank you for the very nice talk. I like the gene modification work that you've done. And we're not as far apart as I thought we were. I think I'll sort of see that in my talk that follows. But one thing right at the beginning, if I can ask, you mentioned MIC is being seen on titanium. Is that really correct? I remember in the 1990s, I spoke with Daniel Pope, who was a pioneer in this area, and he insisted that titanium was never attacked by microbes. So do you actually have any real evidence of titanium?
Oh yeah, this is a very good question. We will have I think a very good paper ready for submission regarding MIC caused by Salmonella and Geobacter against titanium. As we know, titanium rely on their passivation film, which is highly corrosion resistance. Based on our research, we do genetic work on it, and we do TEM and FIB observation from the material perspective. We found two important things. First of all, electroactive biofilm will change the thickness of the passivation film, and then they will change the chemical composition and the structure of the passivation film, which will result in a localized pitting corrosion. And this, with the development of this pitting corrosion, the mechanical property will be significantly decreased because of the passivation film change. But it takes like a month or at least 3 weeks, but with highly colonized electroactive biofilm. But we are ready, like prepare the manuscript, but this work done for 3 years. So now I can say for titanium is now immune to MIC. It's highly welcome for electroactive biofilm.
Okay, I look forward to seeing that when you do get to publishing it, because it certainly was something new
thank you,
titanium to be attacked by MIC. So
Yeah,
do we have any? Yeah, I was going to say, do we have any more questions, Judit?
Yes. So first of all, I absolutely agree with you when you said that there is a gap between the materials scientists, corrosion sciences, and microbiologists. But according to me, this gap is smaller and smaller. So, but what I would like to ask from you, is this Geobacter a heavy biofilm former? Does it form a very thick biofilm in a short time or not?
This is a very good question. It depends on what is the culture medium. When we do MIC test, we want to make material as the sole electron donor for Geobacter. Then we will use a simplified medium we called Professor Derek Lively called a fresh water medium. So the only electron acceptor is fumarate, and the donor is a steady still. Based on this, under this scenario, we can see the biofilm observation morphology for this. They can form biofilm after a day, but they will not form very dense biofilm. I think because of it's a slightly starved environment for Geobacter, so they cannot form a very thick biofilm. But we can clearly see the biofilm colonization on steady steel surface.
Thanks.
Dan Raman Singh here. Do you have time for one question from me?
As long as it's a fairly quick one.
Yes, yeah. Dake, if you go to one of your polarization plots, I'm intrigued that when you have microbial, your potential and current shows high corrosion rate or susceptibility. But after some, when you increase the potential, it stabilizes like it is passivation type of phenomena in the microbial environment. And it was there in other polarization plots as well. Why is that?
This is a very good question. Yeah, I think you can see on this that even though we have biofilm, the coverage for the biofilm is.
Yeah. Okay, Dake. I wouldn't take too much time. And good to talk to you after a long time, Dake.
Okay. Yeah, I think we can discuss it by email. It's a quite good question. Yeah.
Yeah, we have found similar phenomena in our results, and then let's communicate and talk about it. And I better let Dan take over.
Okay. Thank you for your attention, Raman.
Okay. Thank you. Okay. Thank you, Dake. Or Dake, actually, isn't it? Is it?
Thank you, Dan.
Okay. I'm actually the next speaker, so I'll hand over to Judit.
It's my turn. I think I am in a very good position because most of the important factors were already said about you. I mean it about Professor Daniel Blackwood, because he is our next speaker. But I emphasize only some part of it. He is currently Deputy Head of Education and Student Affairs at National University of Singapore, where he has been since 1996, because pretty a nice long time. And the other one, I think it's important, Daniel is a fellow of Royal Society of Chemistry and a member of the National Association of Corrosion Engineers and the International Society of Electrochemists. His current research interests include microbial influence corrosion, corrosion modeling, and pseudocapacitors. So the floor is yours, Dan.
Okay, so my talk's going to be on some of the views that I have on the mechanisms of microbial influence corrosion. I have a different view from the microbiologists, although having just listened to Dake's talk, we're not as far apart as I actually thought we were. So we are getting closer, which is a good thing. So I talked a little bit about, right, we all know how much corrosion costs, so there's $2.5 trillion a year, and it's estimated that 20% of corrosion is due to MIC. so about 0.5% of GDP. But it's not really even an estimate. To be honest, it's much more of a guesstimate. I was speaking to Hans Fleming, who would come up with the numbers first. And he said, basically, it's just a matter of talking to people in industry. If they don't know what the cause of the corrosion was, then they tend to blame MIC. So it's a very, very rough estimate, this 20%. There's no real foundation for it. We know it's caused by a wide variety of microbes, basically fungi, algae, bacteria. Unfortunately, our understanding of the mechanisms are not yet at the level required to control it. So we need more cooperation between microbiologists and electrochemists. I'm working with some electrochemists for microbiologists in our other university in Singapore, Nanyang Technological University, and gradually coming to an agreement on terminology. But there are a certain divergence in terminology. Things like even anaerobic and direct electron transfer, they mean different things to me than they do to my colleagues, as I explain it in a moment. So I'm going to talk mostly about anaerobic MIC because that's economically the most important. But in the beginning, we come up with the problem of what does anaerobic mean? Or anoxic is actually apparently the correct term when it's an environment. You think it's easy, no oxygen. But actually, that's not true. It doesn't take too long if you think about thermodynamics. You come up with your Nernst equation. We've got our term here, log partial pressure of oxygen. You put 0 in that, that's minus infinity. Just doesn't happen. Okay. So when you read reports of 0 oxygen, people often say they've measured 0 oxygen. What they actually mean is it's below the detection limit of their equipment, which is typically, most oxygen detectors tend to be based on a current, right? So they're typically somewhere around 10 ppb. And if you work that out, fixed first law of diffusion, you'll find that 10 ppb oxygen can support about 0.1 microamps per square centimeter. And that's actually plenty to maintain the passive film on stainless steels and titanium. So if you put titanium or some stainless steels in a 10 ppb environment, its open circuit potential will be positive for the reversible hydrogen electrode. So actually there's still enough oxygen in there that essentially there's oxygen at that surface maintaining the electrochemistry. So I tried to come up with a better definition for what I mean by anaerobic. And so what I tend to have is when the cathodic supporting reaction is no longer oxygen reduction, it's probably switched to hydrogen evolution. But then of course that means your anaerobic depends on corrosion rate and the alloy involved. So a piece of stainless steel won't be anaerobic and a piece of steel would be anaerobic. So normally I would do it by measuring the open circuit potential negative to the reversible hydrogen. If it's negative for reversible hydrogen, then I would consider anaerobic. But it's not perfect because what happens if you have other species beyond oxygen and water in there to be reduced? There isn't. So right at the very beginning, we struggle on terminology. So then I'm going to look at some of the possible mechanisms for anaerobic MIC. I'm going to start with the old cathodic depolarization theory. Then I'm going to look at the extended iron sulfide cathodes. Those are the older ones. Then I'll look closer at direct electron transfer, redox mediates, and something on film formation and breakdown. Quite often corrosion rates are dominated by the nature of the film that forms at the surface. So this cathodic depolarization, which has been around for a long time, it's based on the basic idea of corrosion, right? That your cathodic and anodic reactions have to be at the same rate. For every electron transferred at the anode, another must be transferred in the opposite direction at the cathode. So if you increase the cathodic reaction rate, you'll increase the corrosion rate. So that's the basic concept behind the cathodic depolarization theory. And if it's truly anaerobic, then the cathodic reaction is supposed to be hydrogen evolution. So the bacteria consumes the hydrogen at the metal surface, that accelerates the cathodic reaction, and corrosion must accelerate to maintain zero net current. There is some evidence for that. We do see ennoblement. The E-core, when you have MIC, does move positive, usually by less than 100 millivolts, but there's been reports of up to 250 millivolts. So that tells you that the cathodic reaction is going faster. And there are some reports showing only hydrogen consuming SRB affect corrosion rates. But there's quite a lot of evidence against this CDT. If we go right back to what is our hydrogen evolution reaction, we have our Vollmer reaction followed by Tafel reaction or hydroxyl reaction. And we can usually tell which one's dominating by measuring the Tafel slopes. And sort of 30 years ago, Frankenthal and Miller did Tafel slopes for carbon steel and tin actually in seawater. And they found Tafel slopes of 120 millivolts per decade. And they concluded from that that it's the absorption step that is rate determining. So if this was the rate determining step, it wouldn't really matter if you removed hydrogen because you're only going to be accelerating reactions 2 and 3. And so under that, it would say, okay, the cathode depolarization can't be true. However, there's also a slight snag, and I've started realizing that the hydroxyl reaction can also be 100 millivolts per decade if you had high overpotentials. And that's where if it comes on the strength of the iron hydrogen bond, and even here we're not very good. If you Google the volcano plots, which were done by Trezati all those years ago, there's actually 2 types where you'll find the iron now. I tried to find how they were measured differently. Most seem to be based on heats of formation of corresponding metal hydrides. But the older ones have iron on the left-hand side. That means you're going to expect it to be the absorption step. But I have some colleagues who do modeling with DFT, and all theirs, they have it on the other side. And that means it's probably the 3rd step. And so removing hydrogen could potentially accelerate the corrosion. So it's a little, so I used to think that the fact that it was 120 millivolts meant that the absorption step was rate limiting. And so it wouldn't matter whether the bacteria removed hydrogen from the surface. However, that argument is not as strong as it used to be. But it's not the only argument against cathodic depolarization. Okay? This is some data I did years and years ago under waste disposal. In waste disposal, we were always worried about corrosion rates in anaerobic condition, nuclear waste disposal. Right? You're going to bury that waste for 1 million years, so even low corrosion rates are quite important. And what we have here is we're increasing the atmospheric pressure of hydrogen. So you're putting more and more hydrogen pressure. Again, we typically see about 120 millivolts per decade Tafel slopes for the cathodic reactions. But if we go back to our thermodynamics, which is where I always like to start, if it was cathodically limited, we should actually see a shift by 30 millivolts for every decade of pressure of hydrogen increased, basically again from our Nernst equation. But actually what we see in the open circuit potential is we actually see, if we look at E-core, is it's shifting positive. So it's going the wrong way. And also we find as we go to higher corrosion, if we go to higher pressures, we end up with, except for this one, but the majority of cases, we get higher corrosion rates. So that doesn't fit with the idea that the bacteria remove the hydrogen, make the corrosion rate go faster. And I also found that that from weight loss in granitic groundwaters, admittedly that was at 90 degrees, I see, but higher hydrogen actually resulted in higher corrosion rates, not lower corrosion rates. Another evidence against the cathodic depolarization mechanism is in the ennoblement itself. People, like I said, we do see ennoblement. Some typically it's less than 100 millivolts, but some people report really quite large ennoblement, sort of 250 millivolts. The problem with large ennoblement is it actually shifts the open circuit potential positive to the reversible hydrogen potential. So what is the cathodic reaction then? It no longer can be hydrogen evolution, right? So it can't involve those bugs removing hydrogen from the surface. They cannot shift the open circuit potential positive of the reversible hydrogen. And then, of course, there are problems with reports showing only hydrogen consuming SRB affect corrosion rates. There are other reports that show even if it's not a hydrogen consuming SRB, it still affects corrosion rates. So I'm fairly certain the cathodic depolarization mechanism is wrong, but what I'm less certain on is what's correct. Well, this one I was just going to mention it. Another idea has been put forward that H2S is an alternative cathodic pathway for hydrogen evolution that actually involves somehow the H2S molecule. But Nezek actually published something last year that's a modeling, and he was able to show that, all right, in abiotic systems, but in all H2S environments, the cathodic currents can always be modeled simply by just the standard H+ reduction. You don't need some special reaction involving hydrogen sulfide directly. The next mechanism, which I used to like until more recently, is the idea of an extended iron sulfide cathode. Right? The H2S is produces iron sulfide. Some iron sulfides are actually quite conductive, and there can be a better cathode for hydrogen evolution than iron itself. And essentially, we've got galvanic corrosion. This is your iron sulfide, it's your cathode, and iron corrodes over there on the iron. Doesn't work if the ennoblement goes positive for the reversible hydrogen potential, of course. But it worked well for SRB, and I used to be happy. But then people have started finding corrosion for methogens, nitrogen-reducing bacteria. They don't produce any sulfide. And so if there's no sulfide, this mechanism doesn't work. There's also the predictions of electron transfer, but I'll say a little bit more on that in a moment. So if the first question I sort of answer is, does non-SRB anaerobic MIC really exist? And if it does, what is the mechanism? Well, there certainly is a clue in Daker's work, Zhu and Gu, when they showed that SRB caused much higher corrosion rates when they're deprived of easily available energy sources. And it suggested that SRB switched to taking electrons from metal oxidation for maintenance survival energy. I don't quite agree with that, but I am getting closer. So one of the things I wanted to do was to first prove that other bacteria other than SRB do cause corrosion. And we were able to do that with some methogen work that I did several years ago. And over here, we see the weight loss. I mean, it's much higher with the bugs and without the bugs, the methogen. And this was only found when it was starved. If we didn't starve the methogens, we had no accelerated corrosion. And this one here, you see, doesn't seem to be very much higher corrosion rate than the controls. And the reason for that was partway through the experiment, we allowed hydrogen into the system to so basically defeat the bugs again. One of the things I've always found annoying about bugs is they have this habit of dying on you, and that upsets your experiment. You never, so we weren't sure whether the bugs were alive, so we let some hydrogen into them, and then they start producing methane. And as they start producing methane, because they've got plenty of nutrients, they stop corroding. So it was definitely the bugs under stress. They switch to some alternative energy source that causes metal corrosion. However, that's not quite the same as they switch to metal oxidation as an alternative energy source. Okay, there's a slight difference there. They've changed their mechanism, and that causes the corrosion if they're not taking it directly from the metal. This, of course, was always bad news for industry because microbes in the field are under stress. So they're going to be causing more corrosion in industry than they will in your lab. So possible mechanisms. Direct electron transfer. Well, it could be direct contact, right? So our electrons are either going to do direct contact via Pele or via some redox mediator. And when I look at these, I don't believe in direct electron contact. We've got our Helmholtz layer and our electrons jump across there, but typically over about 1 nanometer. Really the heart of electrochemistry, which was Brockquist described it as, is this high electric field that we have at our interface. It's 10 to the volts per centimeter roughly. We essentially have about a volt drop across a nanometer. But biomolecules are bigger than nanometer. You're not going to jump that far. And there's been a number of works that shown that direct electron transfer between aqueous species won't occur over distances greater than a couple of nanometers. And there are very few proteins that we can actually approach the electron surface close enough for direct electron transfer. So it doesn't really seem viable that microbes can. And then there's these pili. Okay, SRB do seem to form pili when attached to carbon steel, and they're a more corrosive environment. But just because you see them in that corrosive environment doesn't really mean that the two are linked, right? There's no cause and effect being established. So if you start thinking, can pili really be electronic conductors over the lengths of microns, then you have to think, okay, what's the mechanism? In papers, you'll see electrical conduction via microbial nanowires, but that's rather vague. I'm quite used to conjugation. We have it in dye molecules in solar cells, which I've worked on before. But this is still nanometers. These molecules are still nanometers in the conjugation. We're not the sort of microns length that you have in a pili. And then there's conducting polymers. Now the conducting polymers, yes, they can be very conducting, but it turns out the really resistant part of a conducting polymer is when it jumps from chain to chain. So the shorter your conducting polymer is, the higher your resistance, because you have more jumps from polymer chain to polymer chain. Once it's in the chain, it's easy for the electron to move along, but chain to chain is actually quite difficult. That was done by Dick Buck in the 1990s. So we have some sort of electron hopping. Again, it's been proposed as mechanism for natural dyes in solar cells, where it jumps from sort of aromatic ring to aromatic ring. But it's still the most resistant step in a conducting polymer. And our short chain molecules that we're going to have in biological systems, they're going to be many orders of magnitude more steps. So it simply turns out it's going to be too resistant. And people have done calculations, and it's better, but it's still only about 10 nanometers. So the likelihood of having conductive pili is it's not impossible, but it's very unlikely. One of the problems is it's actually experimentally very difficult to determine electronic from ionic conduction, especially when your samples are not dry. A lot of the work I've looked at on this, and it turns out that if you do a comparison in the conductivities that are claimed for these pili, it usually turns out to be pretty close to the conductivity of seawater, and which always worries me, right? So you're claiming an electronic conductivity, which is basically the same as the ionic conductivity you'd expect for the media that you've been working in. So then we go to redox mechanisms, and this is where the sort of terminology becomes different. We can either have the electron jump into some species on the surface, or it's got to jump directly into the molecule. Now, which got to get this way. This one, right? You either go via a mediator or it goes directly into the surface of the bacteria. This one, microbiologists call direct electron transfer. And that leads to confusion, because if the species on the surface is some sort of redox couple, it's actually still a redox mediated process. And I could see that really in what Kerr presented, that a lot of what he's actually presented is actually a redox mediator that's bound on the surface. It's not the final product. If it was sulfate reduction, the final sulfate reduction takes place somewhere inside the cell, not on the surface. And that's what leads to confusion. And it's important because if you think of energy terms, there's plenty of energy when you reduce sulfate to corrode the metal. But the energy available for the corrosion is actually the first step in the redox process. So this step to the redox mediator on the surface, that's all the energy you have for the corrosion. You don't have the entire energy from sulfate reduction available. So it does make quite a difference when you're trying to model what's happening. Well, it's also to say that these redox mediators don't need to be in high concentrations because they're going to be continuously regenerated. So when we did the work on our methogens, tried to think, okay, what is this redox species? But it's got to be the oxidized half mediators created by the microbes. So we have the oxidized species is created from the metabolic reaction. We have energy from our anode, our corrosion. Its potential, I never really know what to put in here because, again, you've got the Nernst equation. You have to put something in for the concentration of the ions. I choose 10 to the minus 6 simply because that's what Paul Bay chose for his diagrams. It makes no sense to put in 1 molar because we're never going to get to 1 molar. So we have quite a negative potential for corrosion of iron. What do we know about the cathodic reaction? Well, this oxidized species, which is produced in the metabolic reaction, has got to be reduced again as the cathodic reaction that causes the corrosion. That recreates the minimal energy source. It's got to be positive of Re core. When we measured E core in our experiments, they were sort of up to minus 437 millivolts versus SHE. So our standard redox potential must be positive of that. Okay? Otherwise, open circuit potential cannot be up to that sort of value. The more positive it is, the more energy we'd have to drive the corrosion. Okay, just about could be hydrogen, which actually with the species would be proton. So we could have, it could be that you take hydrogen in the bacteria and produce protons, and then the proton is being reduced. That's not quite the same as the cathodic polarization mechanism because the hydrogen processes occurring away from the electrode surface. Okay. However, when we measure pH changes between starved and non-starved, we don't see any change in the pH. So yet it doesn't seem that the bugs are making protons. However, we're not able to probe the local pH at the surface. If you try and stick a probe through your biofilm, you basically ruin it. So it's still very difficult to measure local pH changes at the surface. So it is possible that the methogens were producing acids when starved. Okay. There are a number of other species that were available that we looked at.
Minutes left.
Okay, I'll try and go quicker. So there were various species that we looked at, and we did find that various ones can cause corrosion, but they're only going to be there in small amounts. You know, so redox mediators certainly can be it. The other mechanism that I think is possible is impact on film formation. Right? We know SRB can make extended iron sulfide cathodes. Could other microbes do something similar? Could we have bugs that make an iron carbide or an iron phosphide surface? We were fairly certain when I go back to this data that we had higher hydrogen pressures caused increased corrosion rates. We thought it probably influenced the nature of the oxide film formed. If the oxide film is less protective, then you will get a higher corrosion rate. But when we looked at the methogen work, we couldn't see any difference in our XRD between the starved and the non-starved. But XRD is not conclusive, particularly as it can only really see a crystalline film. Quite often these films might be amorphous. But there was some work by Valencia and Katerina, I think that is, where they looked at SRB and iron-reducing bacteria together, and they found that that combination gave you 10 times higher corrosion rates. And they found out that the presence of the iron-reducing bacteria, that's iron 3 to iron 2, actually caused cracks in the sulfur film that was forming on the metal. And if you crack your oxide film, then you're going to get, this was actually a sulfide film. If you crack that, you're going to get higher corrosion rates. And that's also what Brenda Little found and Dave Shoesmith when they looked at anaerobic cycling, which always gives you much higher corrosion rates in pure anaerobic. And what Dave Shoesmith thought or found, had some evidence for, was, okay, you make a sulfide film and you let oxygen in, and you'll start making iron oxide underneath the sulfide film, and that cracks your sulfide film. And so once you've cracked that, it's no longer protective and you get more rapid corrosion. So a few conclusions. MIC certainly causes hundreds of billions of worth of damage a year, although we don't really know exactly how much. But when it's hundreds of billions, it's worth doing something about, even if it's 90 billion or 100 billion, it doesn't make much difference. You want to save that money. SRB are not the only culprits. Cathodic depolarized mechanism is wrong, but still don't know what is correct. And until we can come up with a good mechanism, understand the mechanism, it's going to be very hard to produce reliable predictive models. And it's probably more than one mechanism. There's always this idea that we like to hunt for a single mechanism, but it's probably going to be different bacteria cause different mechanisms. And it's always going to be complicated by consortium of microbes in the fields. It's worse when the microbes are under stressful conditions. They seem to switch to an alternative energy source that causes metal oxidation. I'm fairly certain that they are producing some redox mediator that is more corrosive and accelerates the corrosion. We even have some idea that in deep sea it could be sulfur being produced. The sulfur is corrosive. Now, unfortunately, though, industrial situations are nearly always under starved conditions, which is probably why corrosion rates in the field are always so much worse than the ones we have in the lab. So in the future, it's vital that microbiologists and electrochemists learn a common language. I had hoped to go and visit the co up in Shenyang, but unfortunately, COVID's put a stop to that at the moment. And so I'm looking forward to having proper discussions with him again sometime. We've talked before on this issue at various conferences. So once we can understand MIC, maybe we can control and predict it without excessive use of toxic biocides. I mean, biocides do sort of work to stop MIC, but they're not a really very green approach. It's not easy to find a green approach where you kill things, though. So, but I acknowledge my funding, which is from the NRF in Singapore. And I also would like to thank the organizers for helping us have a successful conference. If you'd like any more information, these are some of my recent publications on it. Okay, thank you.
Thank you so much. It was really very interesting. I can't see any questions, but I am really very happy to learn a lot from the explanation of the mechanisms, though I know that it's not very easy to make a decision, either this one, that one, or altogether some of them. Yeah. Do have any person a question? If not, maybe later you can discuss with them. And now and now I give you back the chair.
Okay. It's my pleasure to introduce Iwona Beech, who's also got over 30 years of experience in the field of MIC. That's the third one of us, which tends to mean we're getting older. But you still look young, okay? Mm-hmm. She got a bachelor's in MSc in solid state physics, so she's actually good at mechanisms as well, from the Warsaw University of Technology in Poland. And then she had an MSc in plant pathology and a PhD in microbiology from Imperial and the University of Central London, respectively. She was at one time at Portsmouth. That's when I first came to know her, when she was at Portsmouth. I was at Southampton, which is quite nearby, and many of her colleagues at Portsmouth, Frank Walsh and Sheila Campbell, were also from Southampton. And then she was a research professor at the University of Oklahoma, And she is currently a research professor of the Center of Biofilm Engineering at Montana State, which is in Bozeman, Montana. And her current research is on biomaterials on biofilms on materials, microbe material interactions, biofiling, biocorrosion, biodeterioration, and she's had over 140 publications and several book chapters on the topic. So she's well worth listening to.
Thank you, Dan. So I will talk about the use of metabolomics to look into MIC from the point of view of a possible, finding a possible tool to manage MIC in some sort of, industrial situations. We try to develop this approach from the fundamental point of view and then going into applications. And the industry that we worked with, originally we developed this approach for the marine corrosion, and particularly for the US Office of the Navy Research, ONR. And then it turned out that this approach was also very useful for managing MIC or possibility of managing MIC in oil fields. So when one talks about MIC there are so many different approaches, and in the past decade, obviously, with the onset of novel techniques and progress in both material science, the surface analysis techniques, but also in microbiology, people kind of moved from the microbiological point of view into omics. So metagenomics, transcriptomics, proteomics, and finally metabolomics. And on this slide is kind of depicted what these different approaches can actually offer in terms of information that can be helpful to develop some sort of strategies in terms of an understanding of what's happening in terms of microbial corrosion. We know now that corrosion is a very complex system. And what we're trying to do in Oklahoma, when developing the comprehensive strategy, we thought that the easiest would be to combine two platforms, i.e. the metagenomics with metabolomics. Hence, the two approaches were kind of merged. And we even thought that developing a sort of a big data approach and analysis, combined analysis of this kind of metabolomics pipe or omics pipeline would be very, very useful. However, that has not been realized for different reasons, or not yet. So the total omic visions from the point of view of our approach would be producing a total information-driven corrosion management. And we wanted to ask ourselves, is it realistic? And just I wanted to add that we're still working on this kind of approach. We're collaborating with Equinor in Norway, with Dr. Turid Lingen and her group. And basically what we're doing, we are producing a tremendous amount of data, metabolomic data, from different types of systems that I will just kind of mention shortly, to create this kind of MIC management approach. And then we're hoping that it can be extrapolated to other systems. So not just the oil field installations and pipelines, but also, let's say platforms, let's say also the mooring chains, et cetera, et cetera. So just quickly, metabolomics is the systematic study of the unique chemical fingerprints. So everybody hopefully knows by now what this is, with an onset of metabolomics being so incredibly important in human health. And there's different approaches to sort of individualized medicine based on the individual metabolomes. But recent advances in mass spectrometric analysis or instrumentation help to push this mass spec approach or metabolomics approach forward. So basically, very quickly, what this looks like, so the metabolomics workflow There's basically experimental design, samples and relevant controls. It is very, very important one. We are only as good as our samples are. Then preparation, and of course, it's a bit of a joke because we are using very sophisticated instrumentation, data, and then from this data, we have incredible amount of chemical information in this mass spectrometry, this kind of peaks and this little dwindles there, they are called features, and thousands of features are produced. One can do different type of univariate and multivariate statistics when this data is obtained, and then finally pathway analysis. So rather than having a urine wheel to look at the metabolites like they used to do in 16th century, or the kind of retorts and glassware, we have this all singing or dancing instrumentation, HPLC QTOFs, with different type of bells and whistles. So this biochemical mass spectrometry made tremendous progress again in the past 20 years, and then people are using it very, very frequently. Obviously proteomics, metabolomics, but importantly also chemical imaging. And how would this help us in corrosion? It would basically help us to find some markers or patterns and allow rapid detection, prediction, and mitigation efficacy. So would that approach be a game changer? In our opinion, yes, but it takes time and patience to basically convey the message and demonstrate the power of the approach. So just quickly, we looked at different type of corrosion failures in oil fields around the world, and we chose 4 case studies around the world on different continents in different environments. So North America, South America, and Europe. And this A represents actually Norway, Norwegian oil fields. And then I basically will would like to note that it was very, very difficult for quite a period of time to convey the message that indeed microbes can be present in these pipelines, they can be present in big debris, and then they corrode. Because a lot of times there was no imaging or visual evidence that there is anything indeed in this sort of deposits or pigging material that is produced in these pipelines. Hence, there was a reluctance to acknowledge the existence of MIC in oil field installations. However, you know, this is basically an image that I've obtained, electron microscopy image from the Alaskan pipeline. It was a cookie, i.e. there was a piece of material extracted and cut from the pipe. And then with a little bit of tweaking and different methodology, one can actually, without any fixation and without dehydration and covering the samples with or coating, one can actually reveal the presence of microbial cells in this deposit. So this is just to convince people that indeed bacteria exist, that they are present, and then one can actually visualize them in this material. So the first case study, and maybe I just stop at that study, was the Norwegian pipeline, and very similar physical and chemical conditions, geological formation, seawater temperature. And one system had controlled corrosion, that is basically nothing to write home about, and the other experienced very severe corrosion. So the question was, why? What was the problem with this pipeline? So just quickly, the Norwegian sector in the North Sea, different pipelines, pigging to death. One system has to be pigged very frequently, and the corrosion was pretty dramatic, and versus the other, then nothing really happened. Okay, so the twin line problem was something that we spent some time investigating and also actually publishing. So that publication exists, and one can try to, you know, basically look at it. So we threw metabolomics and metagenomics at it. And then from that work, we progressed further, collaborating with Equinor, to trying to find out what mitigation strategies would result in replacing these microbial communities that causes severe corrosion by the population, or can one do that, to tweak the population and changing the microbial population into benign population, i.e. can one really switch the metabolism and replace the bad players with good actors? So when one thinks about metabolomics approach and compound patterns, well, if we think about traditional chemical analysis, they focus on a few known compounds and abundances in different samples. In metabolomics, we are having tens of thousands of compounds, and majority of these compounds are not identified. So there is a pattern of compounds in the biological systems that are compared. So basically, in any new samples that one obtains, the pattern will give us information on in what kind of state that system is in. Basically, in oil fields, we have, in any basic systems, we have different types of corrosion. We have what people believe MIC, those who believe that there is MIC. We have CO2 corrosion, we have acid, and each type of this corrosion, chemically speaking, will be associated with different compound patterns. So basically, different chemical treatments and other system parameters will also have their own fingerprint on the system. So therefore, one would have to have a tremendous amount and wealth of data and then do principal component analysis on statistical analysis to discern between these different type of patterns that would be related to different type of corrosion attack that occurs, and basically to be absolutely positive that one talks about MIC and therefore develop a mitigation strategy. And sometimes I wonder if this is really that important that we should know whether it's MIC or any type of corrosion, or is it very important just to know that the corrosion is happening, when it's going to happen, and what to do to prevent it. So, so there is oil field metabolome that we refer to, that the crude oil, chemicals, microbial activity, injected water, you know, all kind of parameters that go into this very, very complex systems. And then, of course, metadata is very important, information about past, present, and future corrosion processes that one can discern. So we have microbial growth that may result in low corrosion, like in one of the pipes in the North Sea. We can have microbial growth and very high corrosion, and then corresponding metabolome. We can also have a CO2 corrosion that in this metabolic space that I refer to can be actually differentiated from any other type of corrosion that may occur. So in PCA, or principal component analysis, similar metabolomes are grouped together. Okay, so no matter how many systems we will be looking at, you know, if we can find the similar trend in the systems, in this metabolomic space, we will know that, okay, there is a certain pattern that corresponds to the system, whether it is Alaska or Brazil or Colombia or Nigeria. It doesn't really matter, because the pattern of the chemical pattern of the samples is such that will be indicative of either microbial corrosion or CO2 corrosion or any other type of corrosion. And then for sure, metabolomes will, or metabolomic approach will give that. So then I just skip a few slides, because what happened is that both metabolomes and metagenomes of the pigging envelopes basically showed us that that populations differ depending on the depth of sampling. So the population very close to the surface of the pipe was very different, and metabolome associated with this population was very, very different than metabolome that was collected from the very first pig that was the closest to the bulk fluid, right? So basically what is very, very interesting and very important, that when people sample from these systems, people tend to take a sample and basically homogenize it. And then all the information is lost, because one doesn't really know what kind of metabolomic situation and what kind of population is really responsible for corrosion that one sees in the systems. Hence, what we're doing now, and I've been doing for many years, is sampling in a sense of taking samples from different depths, be that biofilm on steel piling for the low water corrosion, be that the situation with the pigging or any other, always core samples and always trying to dissect it and then compare the populations closest to the surface, because if there is action occurring, it occurs at the surface. It's not, 10 centimeters from, although there are some transport phenomena that may facilitate corrosion from the top down, but typically we should really focus on that, on these interfacial processes. And so very, very important information is could be lost if one homogenizes the samples and does not sample from the surface. Okay, so we have this. What we found in this first shot at the Norwegian studies, it's that we had a very distinct difference in metabolomics, and it was confirmed to some extent by metagenomics between the corroding and non-corroding pipe. So LC is low corrosion, HC is high corrosion, and numbers are designated to pig envelopes. So pig number 11 is the pig that was closest to the metal surface. Pig number 3 was the one that was kind of the third pig that they pushed through the pipe to remove the corroding material and deposits that were accumulating in the pipe. Okay, LC is low corrosion and corresponding pigs from the pipe. So what we could see that they, in metabolomic space, they really, really were very, very different. And also, as it was confirmed later on by more sophisticated data, that high corrosion pig 3 was like day and night from high corrosion pig 11. So what we found is that we had a nitrate reducing and sulfur cycling system. And then for the corrosion, obviously the system moved into sulfate reducing and sulfite producing system. And basically, maybe it was to be expected, but metabolomic changes related to accumulated corrosion, basically was shifting into the sulfur cycle system. And the sulfur cycle system is not just sulfate reduction, but it's H2S production, right? And then associated sulfur cycles. But when one think about it, it's not
wrap
just
up in a couple of minutes.
Okay, well, I don't know if I can. So this high corrosion systems were dominated by sulfate reduction, the low corrosion systems by nitrate reduction. But what is very important is that we then compare the shifts between in this same pipelines when people started treating with trying to prevent corrosion. So a different type of corrosion inhibitors, etc. And what we found that basically we could shift the system by whatever mitigation strategies the people applied, but it was not obvious that the depth of the basically of corrosion, I mean the material, was affected. So basically what all these mitigation strategies were able to do, they were able to change the outermost layer of the corrosion products, but not really going into changing the population or metabolomics or shifting it into low corroding system. So one could, by intervention and by intelligent type of strategies, change the behavior of these populations to make it shifted towards less corroding or non-corroding or just normally corroding environment, but not really when one looks into the populations and materials that were closest to the pipe surface. So then when we compared all the data from all the cases around the world, what we found that the systems that were corroding, they had very similar metabolomes, and the systems that were not corroding or benign or corroding as expected, they all grouped in a certain way. So basically, it was the environment and perhaps material, because obviously there's a certain cast iron that they're using for this production of the pipelines, that determined how the systems behaved irrespective of the geographic location. And it's been reported many, many times. And I've seen it myself, that it was material that was selecting the population. It was material that was selecting the corrosion behavior of this population, not so much, and temperature, that was very, very important, but not so much the geographic location of when this corrosion failure was taking place. And I've seen that on copper nickel alloys, and I've seen that on different types of stainless and carbon steels. So just wanted to show you maybe at the end, because I've got more to show, but obviously run out of time, is that one can then extrapolate and go into metabolomic pathways and basically see what happens in terms of at least the first shot, and what is the kind of metabolome that one looks at, is which were the pathways that were basically common for all these 4 locations worldwide in terms of a production of the chemicals that were commonly found. And then from there, one can obviously build on trying to find corrosion or biocorrosion markers. So when one starts seeing accumulation of a certain type of compound, it could be, or compounds rather, because it will be complex and will be more than one for sure, then it will be maybe a warning sign saying, okay, it is to be expected that maybe the onset of MIC will take place. And then one can do this by looking at different types of corrosions, not necessarily microbial corrosion, but chemically induced corrosion or chemically driven corrosion, abiotic corrosion in a variety of environments. So I understand that I probably overstated, extended my talk, but there is also another technique that we were using, and this is imaging, chemical imaging, that is incredibly, incredibly important, because we can actually find and match the type of compounds that we're seeing in situ on the surface with corrosion damage that happens underneath a biofilm or a corrosion products. So, so basically laser ablation, mass spectrometry, and analysis and imaging in situ can really give us a very important information about the biology involved and the type of chemistry that is involved in corrosion. Thank you very much. I think this is probably my time is over.
Okay, thank you. Maybe I will start with a question.
Okay.
I like your idea of replacing the corrosive bacteria with benign ones. It sort of almost seems like, well, why didn't people think of that before? But also it seems that it's difficult to replace, once the bacteria are on the surface, difficult to replace them. So would it be worth maybe seeding right at the beginning with benign bacteria?
Well, this is a problem, because there is obviously evolutionary there is a selection pressure, and it all depends on, even if we seed it with so-called good bacteria, the problem is maintaining the population in the state of being benign. And so that is one big mystery in microbiology. There is a microbial warfare going on all the time. So basically the bad players want to take over. And this is incredibly complex. There's not that much information that have been coming from that research that's been used in industry, that is mainly for medical fields. But what happens is that once we were thinking about seeding, and people have demonstrated that even if you seed with something that could be protective, very shortly or with time, the system will change. Okay, I think that that maybe the most important thing is switching the metabolism. So these good players and bad players, in my view, bad players can become maybe harmless if they are switching into different electron acceptors or electron donors. So if we starve them of electron donors, we will then maybe emphasize that the population, because it's a very complex population, right? We have both bad and good guys. So to allow the good population to take over and suppress the bad population. These attempts were made by obviously.
I think I better stop you there, Iwona,
yeah.
because we're running way into the next
Sure.
talk.
Okay.
Okay, thank you.
No
It was a
problem.
very, very
People want
interesting
to
talk.
ask many questions, but yeah, but people tried nitrate treatments, etc., and it worked or didn't work for different reasons. But yeah, there are strategies, of course.
Okay,
Thank
I'll
you.
hand it back to Judit.
So thank you. So now our next speaker is Dr. Kaushal. He is an assistant professor of instruction and has earlier served as a postdoctoral research associate at the University of Texas at Arlington. He has taught a variety of undergraduate and graduate courses in construction energy and management and general civil engineering. He has 5 years of experience working in the field of construction, management, research, and teaching. He has authored more than 50 peer-reviewed journal and conference papers. He has been actively involved in the industry research projects on sustainable trenchless pipeline construction and renewable technology. He is a member of the American Society of Civil Engineering and Cured-in-Place Pipe subcommittees, and he's also a member of the Early Career Editorial Board for Elsevier's Underground Space Journal and board member of MDPI's Infrastructure and Sustainability Journal for his expertise in sustainable underground infrastructure. The floor is yours. Please try to keep the time because we are a little late.
Thank you very much. Thank you for a very nice introduction, and I appreciate your time and the opportunity for me here. Very good afternoon to one and all here, and thank you very much for giving us time to present. And the topic of my presentation is microbially induced, or sometimes it's called as microbiologically induced corrosion of concrete in sanitary sewerage system. So I'm going to talk a little bit of the general processes involved in this and some of the control mechanisms which are associated with this process. I know that there are experts all over the world, so some of the things would be kind of repeat, but still, since the scope of the paper, and we are going to update the paper we are working on related to this, so far as the literature and the other things are concerned, but just this presentation gives an overview. So along with me on this paper, we have Dr. Najafi, who is professor and director at the Center for Underground Infrastructure, Research and Education, which is CURI, at the Department of Civil Engineering, the University of Texas at Arlington, USA. So the outline of my presentation, I'm going to go with the introduction and background of MIC, the objectives. So I'm going to talk about how MICs occurred, basically in the sewerage environment, and some of the historical attempts which have been made to prevent MIC. I'm going to talk about antimicrobials, some of the MIC and repair by antimicrobial, what are the cost implications associated, the laboratory testing procedure which was undertaken by us so far as for the scope of that project. So what were the standards followed, and of course the results, conclusions and recommendations for future research. So as you all know that MIC, which is microbiologically induced corrosion, it's also called as hydrogen sulfide corrosion, is basically caused by the production of hydrogen sulfide by the sulfur oxidizing microorganisms which are present inside the sanitary sewers. and is also the main source of concrete deterioration in the sanitary sewer structures. So over this past 70 years, or now is more than that, there have been attempts to fortify the concrete against this direct acid attack, which have proven to be fruitless. So there have been a continuous research and significant amount of research done with this regards to understand the complexities which are involved in these mechanisms. So the objectives of this presentation, this study, is to understand the process of the occurrence of MIC by hydrogen sulfide, and to kind of recommend some of the solutions or some for the reduction or the mitigation of MIC. So as you can see here in the picture, the dissolved hydrogen sulfide is first generated in the anaerobic sulfate reducing bacteria. You can see here. So these bacteria basically, it colonizes in the slime layer beneath the surface of the flow, and to create hydrogen sulfide as a byproduct as it begins to break the organic matter. So now the hydrogen sulfide gas is generated when first the dissolved hydrogen sulfide is stripped from these flows. Basically, you can see the flow here, and also at the same time there is a turbulence such as force mains, and also the drop structure is basically introduced, right? So then we can see the release also from the laminar flows, right? The wastewater which is going through different structures, different hydraulic structures on this way going. And when the dissolved hydrogen sulfide concentrations become elevated due to the prolonged retention time, so as the time increases, there is a increase in the concentration of hydrogen sulfide, because this is a continuous process and this keeps on happening with the passage of time. So further we see when the hydrogen sulfide gas is released, the sulfur and sulfates, they are placed on the surface of our structures, which here are the sanitary sewer structures, right? And now we would prefer this process to just stop here, but no, it further introduces yet another bacteria. We'll see how and what to break down the byproducts that were formed previously. As you can see here, these are some of the radicals. So this is where the hydrogen sulfide corrosion actually begins. And because their bacteria is Thiobacillus, which actually takes into play, the acid is actually produced, which is the sulfuric acid, right? So now you can see here when this bacteria, which is Thiobacillus bacteria, it begins to metabolize the sulfur and sulfates, which now comes from the concrete environment, because usually we have seen the structures which kind of are susceptible to this issue usually are corrosion, concrete structures, from where now the sulfur and sulfates are going to come, and which in turn produces the hydrogen sulfide gas, and which in turn produces the sulfuric acid. So this was a kind of process which is a very general one. So the historical attempts have been made in the past to prevent and present this issue of MIC. So as you can see here, the barrier coatings in form of polymeric linings, which could be made of epoxies, polyurea, polyurethanes. So as you can see, different, now there are several industry products which claim to be very effective or close to 90%, 95%, or even more than that. But still there are some challenges which are involved, or there are some environmental impacts in terms of styrene, etc. So that also the carbon footprints got to be taken into consideration so far as the prevention of MIC is concerned, and the use of these polymeric linings. So another type of method which could be used to prevent or to mitigate the issue of MIC is the use of antimicrobials in addition to these polymeric linings. So the concept of antimicrobials is such that when they are introduced in the very beginning of the development of structure, which is a hydraulic structure, concrete structure, so this antimicrobial is added to the ready mix, right, concrete. So the idea is to kill the bacteria that basically colonizes on the exposed concrete surface, and breaking the chain of this occurrence of MIC that basically produces sulfuric acid. So now how it's going to act is this way. So as the Thiobacillus bacteria attempt to colonize the antimicrobial treated surface, they are met with this electrochemical shock with the help of this antimicrobial. And therefore, if there will be no bacteria, there will be no acid produced, and as a result of which there is no corrosion. Now, as you can see in this slide, we have a concrete wall box which was affected by MIC, as you can see here, this corrosion on the concrete surface. And then as a result of which this antimicrobial was used. And the good thing about that is that it could also be used on like treating the walls of the hydraulic structures. So you don't have to rebuild those in the form of coating, right? And also, of course, in the very beginning. So now you can see figure C, which shows how the antimicrobial mix was treated, and as a result of which there is a significant decrease or at least no corrosion which can be seen, and we can get rid of this issue. Now the thing which comes into play is the cost. So we have been seeing that if we did a little bit of market research all over the US and North America in general, like Canada and other places. So precast, when we use it, so basically it ranges from $75 to $95 for 4 feet diameter. And so far as which is a vertical foot, needs to be paid attention to. And for the cast in place per cubic yard, it ranges from like $140 to $175. And so far as the rehabilitation, we are not talking about the new construction, but just to rehabilitate or renew the existing surfaces or structures ranges from like $60 to $90 per vertical foot for 4 feet diameter structure. So in addition to this, there is also need to specify antimicrobials. So why do we do that? Is to eliminate the bacteria that causes MIC or hydrogen sulfide corrosion, to kind of provide a permanent relief from this issue and protect the structures, and also to provide a versatile solution with the precast, cast in place, dry gund, and rehabilitation of pipe and structures, and also to provide a surface that will not wash off or abrade or leach and pinhole, and also to eliminate the delamination, etc. So the testing procedure which pretty much was followed was as per ASTM D4783 method. So basically, the concrete samples, 2 samples were taken, and they were added the antimicrobial additives. Now we don't know initially in which one it was added and which one not. So in one it was added, second it was not. Now the samples, they were carefully conditioned to reduce the surface pH and also the alkalinity, and a cell suspension of Serratia mescens was prepared and estimated at 10 to the power 7 colony forming unit per milliliter. Okay, and the suspension was placed on the surface and allowed to dry as per the standard suggested, and the samples were kept for like 24 hours. So then it was further made to perform a cell recovery method, and then the plates were like re-incubated for 48 hours. So after 48 hours of incubation, it was very interesting to see that there were light pink colonies on the surface of samples, which indicated that there were no antimicrobials added. However, the sample in which the antimicrobial admixture was added, there was no growth of colonies. So as you can see here, sample 1 and sample 2. So after 48 hours of incubation, sample 1 had light pink colonies, as you can see, and sample 2 showed no growth, which shows that the antimicrobial acted against the building up of the colonies, which would have led to the corrosion. So after 48 hours of incubation, the antimicrobial chemical showed resistance to the growth of the bacteria. So in the conclusions, I would like to say that the formation of hydrogen sulfide and its escape from the sanitary sewage environment is essential for the MIC in these sewer structures or systems that do not lead to the development of corrosion. Also, there is a considerable biological activity in the sanitary sewer collection systems, which is a complex phenomenon and which produces sulfuric acid. And also, as we have seen in the previous slides, there are presence of calcium-based minerals, which usually comes from concrete as a binder phase, and which is kind of, it becomes hard to tolerate the corrosive sulfuric acid. And compared to the polymeric treatment or coatings such as polyurea epoxies and polyurethanes, the use of antimicrobial proved to be kind of more effective in the elimination of MIC. And the sulfate reducing bacteria under anaerobic conditions in the submerged part of sewers facilitated reduction of sulfate ions and production of various forms of sulfides, which kind of eliminated the production of the sulfuric acid. And the testing was done as per ASTM 4783, which showed the resistance of concrete samples to this microbial attack with the use of antimicrobial additive. So in the recommendations, I would just like to say that there is a need to model the hydrogen sulfide production and accumulation along with the corrosion rates, which I have seen. Yes, I'm just getting to there. So, which basically there is a need to do that and to study, and I have seen present studies like in 2020-21, they are the authors are researchers are attempting towards that, and so there is a need of continuous modeling. And as the deterioration of concrete is a complex process that involves varied surface interaction, there is also a need to better understand the other empirical inputs, as the previous author was talking about the various factors which involve the MIC production. And then also there is a need for the detailed examination of the specific points where the corrosion begin and begin to migrate the microbial community along with the concrete surface is also pretty much needed. So I'm open to questions and we'll be happy to address any. So for more questions or feedback, please reach out to me on my email address, which is here, and or our center. You can reach out, so you can visit this name and say UTA, University of Texas at Arlington, and you should be able to navigate through. Thank you very much. Thank you for the opportunity. I really appreciate it.
Thank you so much, but I'm really sorry, but there isn't any possibility to have any question. I'm sure that who has any question can join you through the emails. Thanks for you. And now
thank you very much. I appreciate it.
And now I give the task to Dan.
Okay, our next speaker is Katerina Kreislova. She graduated from the University of Chemistry and Technology in the beautiful city of Prague, and she now works as a senior scientist at the SVÚOM Limited, a company that specializes in the field of corrosion protection against corrosion and surface treatment. She's a researcher in the material protection area. Her specialization is in atmospheric corrosion, and she's particularly interested in basic mechanism and kinetics. She is involved in the ISO TC156 RWG4 group for atmospheric corrosion classification, but she's actually interested in all corrosion problems that are related to industry. Okay, so let's welcome Katerina.
So good afternoon, everybody, and let me show some examples from the practice, how we try to evaluate if the corrosion which is occurred on the pipeline on another system is connected with the microbiologic induced corrosion. So after the very long afternoon presentation, I cannot go in deeper. What does it mean, microbiological induced or influence corrosion? Just to remind that may be connected with a very different type of corrosion. We can identify the pitting corrosion, crevice corrosion, and to identify if this type of corrosion is this mechanism is joined, is influenced by the microbiology is very difficult, because in many cases we are coming to this system, to this pipeline in the case where are leakage, where are penetrated, and this is problematic in a very heavily corroded surface to identify the microbiology. So what is the system when we met with this type of corrosion? It's different cases. It's a drink water treatment plant, cooling water systems, heating water systems. Very frequent now is a corrosion in fire protection sprinkler system and also in the sewage water treatment plant. The typical metallic materials in which the corrosion is occurred is stainless steel. Usually it's a type 304. In very specific cases 316, but in mostly it was 304, and carbon steel. But we can find this type of corrosion also on galvanized steel, brass, and other metals. How we can identify the presence of bacteria in the system? We used three basic systems. The first is that we're looking on the water which is in the pipelines on another system, which means we perform the basic chemical analysis, looking for the sulfate, looking for the other species, and also the basic bacterial tests to identify the presence of iron or sulfate reduction bacteria. We also look on the corroded surface and looking on the morphology of the surface, the character of the pits, and perform the detailed analysis of corroded surface to identify a higher amount of the sulfate or of another species. What is very typical, very characteristic for the cases of microbiology-induced corrosion is existence of presence of such dendritic skeletons on surface, which is very characteristic. It's a residual of biofilm which created on the metal surfaces. And we also, as I mentioned, that we're looking for the concentration of bacteria. We tested the amount of colonies, and this is some limits which give us information how corrosive is the system. The analysis of surface give us information about the presentation of bacteria and on the sulfur in the surface. This is the first case study. It was a pipe of carbon steel in a cooling water system after 3 years in service, and the information about the water show that is not so high concentrated, but the concentration of iron bacteria was relatively high. And you can see how looks the corroded surface. That is a lot of turbulence on the surface, and after cleaning the surface, it's evident relatively shallow pits, which was full of sulfur. The second case was also from the carbon steel. This is the pipeline from sprinkler system. It was the main horizontal pipe, and especially in the lower half of pipe, it was occurred a lot of voluminous tubercules. After their cleaning, we can find that this lower part of the tubercules is very different color. It's a non-homogeneous heterogeneous layer which was created by the Judit and lepidocrocid, and in the black part, which is near to the steel surface, was also identified magnetite. The sulfur content in this layer, in this tuberculous, was near 2%. This existence of such tuberculosis is usually connected with existence of iron oxidation bacteria in the water system. And we look very deeply on the layer of corrosion products. We can identify the layer of biofilm, the layer of oxide, and also the layer of sulfate and the sulfates. So you can see how different is the morphology of the surface. The case study 3 is stainless steel. After 5 years in service, it was a cooling water system. In this system was a relatively high content of the sulfates, and we identify a relatively high content of both the main type of bacteria which is connected with the microbiologically induced corrosion. On external surface of the pipe was such places where identified the penetration, the leakage, but on the inner surface was a relatively homogeneous layer of biofilm, and after cleaning it was identified a lot of pits. We look on the morphology of the pits, which are also very typical for the microbiology corrosion, and on inside the pits we identify high amount of the sulfur, opposite to a layer of the other surface which was not corroded and where is the content of the sulfur relatively low. The case study 4 was heating exchanger, which was used for the cooling water, also stainless steel tubes. And the problem is that all these relatively small pipes is joined together in such as spirals. There is a very narrow space between each one, and there is accumulating a lot of biofilm. It was still such a vasky layer, typical for the biofilm, and after cleaning, it's evident the line of small pits which was just occurred in this crevice between the pipes. This biofilm we tested on the concentrate of the existence of the presence of the some bacteria. It was not very successful, but it shows some existence of, again, iron reducing bacteria. The characteristic pits and the morphology of residua was again typical for the microbiologically induced corrosion, and the content of the sulfur was relatively high. Case study 5, it was sprinkler system. After 4 years in service, the pipelines was stainless steel. Again, it was a water storage tank, which is used as a reservoir for the water for the system, and we can identify a lot of iron reducing bacteria. In the pipeline, there is a lot of pits, the characteristic, and it was identified about the 16% sulfur in the corrosion products, and after the cleaning the corrosion products in this residual biofilm structure was also identified about 5% of sulfur. In the system, we also now find the brass fitting, which was heavily corroded, and it was also very typical characteristics of pitting and selective corrosion of the brass connected with this type of corrosion. Case study 6, it was the drinking water treatment plan, also stainless steel. It was interesting because the first penetration occurred after 6 years of service. The pipeline was repaired, and in this repairing place, the penetration occurs just only after 1 year of the new materials. That means that if the pipeline is new, it takes a relatively long time for the bacteria to colonize the surface. But just in the case it's a made only local repair, this place is very quickly colonized by the other bacteria from the other inert surface of the pipes line. Here we can identify a lot of bacteria, both of them sulfur and iron reducing bacteria. Case study 7, also the drinking water treatment plan, again the stainless steel. The penetration occurs in 2 years in service, and you can see how looks the morphology of the surface. The content of the sulfur was not so high, but the presence of bacteria, especially the iron reduction bacteria, was high. And the last one is the case study 8. It was the pipeline in the sewage water treatment plant, stainless steel, 2 years in service in weld area, where is inside surface is a very poor passivation. The such corrosion, such pitting corrosion occurs, and again we identify a lot of sulfur in this characteristic morphology of the surface. High content of the sulfate was identified in water, and very, very high content of the bacteria was identified in the water. It is not surprising because it is a waste water. When I summarize all these case studies, it was interesting to show that what is typical for all these cases, that the penetration of the material of the pipe wall occurs in a relatively short time. One expression is that this sprinkler system, but it is due to the relatively thick wall. Other typical is high content of the sulfur in the corrosion products. The water analysis was not so can be used as a prediction of this type of corrosion, because it's very different. And what was also very surprising, that in many cases we cannot identify the sulfate reducing bacteria in the water, but in practically all cases we identify the iron reduction bacteria in the water, except again this system, but we have not water sample to be analyzed. So it's very, very typical for this all cases, and we can conclude that in cases of such a system is very difficult to make some prediction about this type of corrosion, because we usually solve this problem after the penetration of the pipeline, after the leakage of the system. We are not, in many cases, we are not able to solve the problem immediately, and sometimes we obtain the samples of the pipe after some days of some weeks from after the withdrawal them from the system. So this is a problem to find some living bacteria on the surface. So we need to indirect method for the identification if this premature corrosion is caused by this type of corrosion. So it's a one crucial problem in the industry. How when we think and try to identify if the corrosion was caused, was accelerated by this type of corrosion, we need to complex analyze of water, material, surface, corrosion products, and all these techniques which are available to identify and put together this mosaic to be sure if this corrosion was caused by the combination of environment and microbiology activity of the water. So what was necessary, and it was mentioned in some previous presentation, that we need to apply the new identification techniques, but in a more simplified way, to be applied not only in the high equipment laboratory, but to be able in the field for the easy identification, and especially for the protection of this system. The first step is necessary to analyze the water source. If this risk of this type of corrosion is necessary to do some protection against this type of corrosion. So I would like to conclude with the sentence that the most challenging aspect is of this type of corrosion is lack of predictability. So thank you for your attention.
Thank you. And unfortunately, we don't have time for questions because we're already running into the next group's time, and we still have one talk to go. But if people have questions for Katerina, perhaps they can send it to her via the chat or the Q&A, and she can answer it via that way. Then I will go back to my co-chair, Judit, to introduce our last speaker.
Yeah.
Okay.
Nataliia Tkachuk is Associate Professor at the Department of Biology at the Shevchenko National University of Ukraine. She has got the MSc degree in teaching chemistry and biology and the PhD degree in microbiology on the role of microorganisms of polluted soils in deterioration of steel. She had teaching activity from 1998 till the present time in the topics microbiology, virology, immunology, ecology of microorganisms, and biotesting. Between 2004 and 2009, she was associate professor in water chemistry, microbiology, and general and inorganic chemistry. Her scientific interest is the materials biodegradation, biofilms, sensitivity of organic compounds of bacteria, active in biodeterioration of materials, and so on. She is co-author of 120 scientific publications and participated at 70 scientific conferences. Nataliia, now it's your turn.
Thank you. Hello, everyone. I would like to sincerely thank the organizers for the invitation to speak at this conference. It is a great honor for me to have the opportunity to address such a distinguished audience. The title of my report is Biofilms of Sulfate-Reducing Bacteria on Polyethylene Terephthalate, the effect of bacillibactin-producing strains of Bacillus valenciensis. Biofilm is a surface model for the growth of microorganisms. The role of biofilms is being studied in medicine, wastewater treatment, biodegradation of toxic compounds, biodamage of materials. Due to the high biochemical activity and the ability to form biofilms, bacteria are actively involved in the degradation of polymeric materials. A significant amount of polymeric materials is represented by polyethylene terephthalate, PET, the production of which is growing every year. This is the structural formula of polyethylene terephthalate. Plastic PET bottles are the most common type for packaging soft drinks and water. In Ukraine in 2018, out of 203.7 thousand tons of PET bottles that become waste, only 50,000 tons ended up in waste processing plants, about 25%. The other 75% were buried in landfills. In addition, due to the low level of environmental awareness and education of the population, a significant part of the used PET bottles does not end up in landfills, but in natural ecosystems. Polyethylene terephthalate is very resistant to degradation by microorganisms. The ability to degrade the PET is inherent in a small number of species. For example, Bacillus cereus, Bacillus hotellii, Dionella sacchaensis. Among the microorganisms isolated from the plastic surface and biofilms, there are representatives of sulfate-reducing bacteria of the genus Desulfovibrio, Desulfobulbus, Desulfatitolea. We used Desulfovibrio oryza as a model organism to study biofilm formation on PET. Also, they did not have a degrading ability with respect to PET, according to the results of gravimetric analysis. Bacteria Desulfovibrio oryza were identified, which are participants in the damage of metal construction in soil. You can see the electronic microphotographs of Desulfovibrio oryza, magnification 22,000. A number of researchers point out that ferrum 3+ plays an important role in the formation of microbial biofilms, and the depletion of iron in the environment can slow its formation. Siderophores are low molecular weight compounds that halate ferrum 3+ ions, converting iron bound to proteins or water-insoluble compounds into the ionic form of ferrum 3+ available to microorganisms. Many aerobic and facultative anaerobic microorganisms are able to synthesize siderophores, the presence of which changes the structure of the microbial community. Antibiofilming properties are known for Bacillus valezensis bacteria as their ability to produce siderophore bacillibactin. Siderophores are ferrum 3 halators, and halating agents affect the stages in biofilm development. The ability to form bacillibactin is characteristic of Bacillus valezensis, NUCC1, and NUCC2b. You can see on this photo Bacillus valezensis strains and microphotographs of the cells NUCC1 and NUCC2b. To study the intensity of biofilm formation on the PET surface, 10 by 10 millimeter samples of PET were cultured in post-Gates C liquid medium, 76% of the volume, with the addition of mid-peptone brose OSU pernatant from mid-peptone brose cultures of Bacillus valezensis, NUCC1 or NUCC2b, obtained after centrifugation for 10 minutes at 5,000 round per minute, 22% of the volume, and a culture of Desulfovibrio oryza, NUCC-SRB1, 2% of the volume during 50 days. a biofilm assay, indirect measurement of bacteria biofilm biomass by crystal violet adsorption-desorption was used. Statistical analysis of the obtained results was performed using the statistical model of Microsoft Office Excel 2010. Determination of antagonistic properties of Bacillus thulesensis, NUCC-C1 and NUCC-C2b against sulfate-reducing bacteria. The method of filling bacterial colonies of Bacillus thulesensis with agar medium containing the test organisms, the Sulfovibrio oryza, was used. For this purpose, daily cultures of Bacillus thulesensis strains were sown with a Drigalski spatula on the surface of meat pepton agar in petri dishes to obtain separate colonies. After the colonies of microorganisms were well developed, around the colonies that were grown, Postgate C agar medium was poured with other test organisms, sulfate-reducing bacteria, the Sulfovibrio oryza, 10 raised to the 6 power cells per milliliter medium. After solidification of the medium with test cultures, anaerobic conditions were created by the method of Sturm in the modification of Duda. The development of bacterial test culture was visually observed. It was found that the supernatant of the studied Bacillus thulesensis strains significantly inhibits through time the formation of a biofilm on the surface of PET bacteria, the Sulfovibrio oryza, NUCC-SRB1. It should be noted that the inhibition of sulfate-reducing bacteria biofilm formation on PET by the supernatant of both strains was the same, despite the observed different level of expression of Bacillobactin siderophore synthesis genes and the difference in the number of catechol type siderophores. Such a supernatant from mid-pepton broth cultures of Bacillus thulesensis, NUCC-C1 and NUCC-C2b inhibits the formation of sulfate-reducing bacteria biofilm on the polyethylene terephthalate surface. Therefore, we future investigated the antagonistic properties of our Bacillus thulesensis strains against sulfate-reducing bacteria, the Sulfovibrio oryza. It was found that sulfate-reducing bacteria, the Sulfovibrio oryza, develop around colonies of strains. Sterile areas are absent. Therefore, the antagonistic properties of the studied strains Bacillus thulesensis against sulfate-reducing bacteria were not observed. Thus, we can draw such conclusions. This therefore producing Bacillus thulesensis strains inhibit the formation of sulfate-reducing bacteria biofilms on the polymeric material polyethylene terephthalate with its long-term exposure 50 days in a culture of bacteria under conditions of sufficient iron supply. Antagonistic properties of the studied Bacillus thulesensis strains against sulfate-reducing bacteria, the Sulfovibrio oryza, were not observed. Bacillobactin-producing strains prevent the development of bacterial biofilms on the polyethylene terephthalate surface. This is one of the reasons for the prolongation of the process of polyethylene terephthalate by degradation in natural ecosystems. Thank you very much for your attention.
Thank you, Nataliia. First of all, you helped us because we ran out of time and your presentation, you kept it time. So maybe there could be one question. I haven't seen any of the questions, but maybe I ask all those participants who would like to ask something from Nataliia, please keep in touch via email. And now, as we have ended this section, first of all, I would like to express my thanks for all presenters for the group presentations. It is very good that within this web conference, the topic MIC was presented by not only academic, but from the industrial part, which shows the importance of this type of corrosion. So thanks for your help. Thanks for Daniel.